everyone, today I'm going to present a summary of the article called A Fluorescence in Situ Hybridization FISH Microfluidic Platform for Detection of HER2 Amplification in Cancer Cells. So basically, as an introduction, in gastric cancer, patients can significantly improve their overall survival rates by taking the targeting drug Herceptin, which directly targets HER2, a protein overexpressed in this type of cancers. Therefore, a highly specific and accurate detection method is needed to prevent unnecessary medical costs and drug abuse. Fluorescence in situ FISH hybridization is an important technique with high specificity for localizing mutant genes or their respective mRNAs within individual cells, widely used for detecting HER2 amplification. Unfortunately, it is a labor-intensive, time-consuming, and costly process that also needs well-trained personnel. So, in this work, a new integrated FISH microfluidic platform aimed to automate the entire FISH assay for the detection of HER2 by using biomicroelectromechanic systems, known as biomemes, technology is proposed. This technology is capable of producing micro-devices comprised of low-cost materials that are associated with lower degrees of contamination due to their usage of lower reagent volumes and their disposability to a single use. Moreover, the miniaturized microfluidic system developed by biomemes have demonstrated similar or even superior performance to their large-scale counterpart. So, as part of the methodology we can see here, first they use 4 breast cancer cell lines and 2 clinical tissue samples, including non-HER2 amplification cases as negative controls and also HER2 amplification cases as positive controls, demonstrating HER2 overexpression. Also, fixed cells on glass slides were used to perform on-bench and on-chip FISH analysis. For FISH microfluidic chips design, we can see here the design. 2 major microcomponents were produced. Basically, the micropumps and the microvalves here that are normally closed. Basically, so when the air channel above the micropump is manipulated, there is a thin membrane on the micropump that will be open or closed, so the reagent fluids can be transported to the reaction chamber below the micropump. So, in this case, for example, for the FISH analysis of cells in a chip, they perform the next protocol. First, they use a 2x sodium chloride sodium citrate buffer, SSCC, with a pH of 5.3, heated to 73 centigrades. And it was first injected into the reaction chamber while activating the micropumps. Then a treatment with 0.0025% pepsin was administered to digest the membrane proteins. After using PVS for washing, there was the fixation with 1% formaldehyde, and the cells were then sequentially dehydrated for 1 minute using ethanol. In this case, they use 70%, 85%, and 100% ethanol. Then, in the next part of the chip, a 2 microliters probe solution containing an orange fluorescent probe specific for the HER2 gene and a green fluorescent probe specific for the chromosome 17 centromere was transferred to the reaction chamber. As we see here, we have a different temperature. So the DNA here were denatured at 75% and 75 degrees for 5 to 10 minutes, and then hybridized for 16 hours in the other part of the chip at 37 degrees. Then, after washing, the DAPI solution was used to stain the nuclei. And after the whole process was done, fluorescent images were acquired. This is in the case of just cells, but for FISH analysis of tissue chips, they first use a paraffin-embedded tissue, sliced and placed on glass, and slided below the reaction area. It means under this part of the chip. And then the protocol was similar to that used with the culture cells, there was a sodium thiocyanate used instead of SSC, and double distilled water was used instead of the PVS. Here we can see how is the actual chip managed. and we can see that it has 3 layers. In A, we have an air layer, B, we have a liquid chamber layer, and in C, there is the glass slide. It's important to mention that the air layer and the liquid chamber layer were made from PDMS. So, as a result from these experiments, we can see how, in order to automate the entire FISH protocol, a suction-type pneumatic micropump acting in concrete with microvalves played a key role in the microfluidic chip designs, which is here the micropumps and the micropumps. Basically, the deflection of the PDMS membrane below the micropump here was created by a negative gauge pressure in the air chamber, which provided a driving force to make the fluid flow through the microchannel. Therefore, the performance of the micropump was dependent upon the volume transported and the back pressure. The pumping rate against the applied air pressure is shown in the next picture. Here we can see how the maximum pumping rate of the micropump integrated on the chip was measured to be 35 microliters per second here, at a gauge pressure of minus 50 kilopascals and a duration of 12 seconds per cycle. Also, as expected, the pumping rate increased with the applied gauge pressure. The higher the air gauge pressure was applied, the bigger volume of the PDMS membrane was created. So, however, at a high gauge pressure, such as minus 50, minus 60, and minus 70 kilopascals, the pumping rate reached a saturated value and had no significant difference since the membrane was deflected maximally to touch down the channel. Next, checking the results of the FISH analysis, we can see here how they use both 2 breast cancer cell lines, MB468 and MCF7, as negative controls, as they don't show HER2 overexpression, and SKBR3 and MDMB361 as positive controls, as they present this overexpression of HER2. How they measure it? Basically, HER2 genes were depicted in orange, how you can see here, while the chromosome 17 centromer genes were green. And HER2 overexpression was defined as the ratio between the number of fluorescent spots associated with the 2 genes. And also then a quantitative fluorescence data were compared between the traditional benchtop protocol that we can see in the image of the left and the obtained from the developed microchip that we can see in the right. So first, in figure A, we can see figure A and B are the negative controls, as the ratio between both genes are balanced in the benchtop and the microchip. In the figure C, it's shown that a single cell is characterized by more than 6 CEP17 genes, which indicate in both cases polysomy. And in image D, it is a typical status of HER2 overexpression. And in E, we can see the same results, but the detection of HER gene amplification successfully made it in images of tissue samples. Here we can see also the statistical analysis made between the benchtop and the microchip assay, and we can see that in the p-value comparison is non-significant. So basically, as a conclusion, here was presented for the first time a microfluidic chip capable of conducting the entire FISH process in an automatic format for the detection of HER2 amplification in breast cancer cell lines and gastric clinical tissues within 20 hours, with 70% reduction of rearrangements when compared with the traditional benchtop protocols. Thank you for your attention.